Good afternoon, this is Mrs. Kantz, and this is the review video for Unit 5 test on molecular genetics. As we go through this information, you are able to fill out your review sheet so that you can study from that review sheet. All of these questions are topics pulled from the test. The goal in being able to give you this review is so that you can study this information as well as some other information if you don't understand this information, be able to go back to the content, and then be able to complete the Unit 5.12 test and be able to score above a 70%. So let's go ahead and get started. Your question number 1 says, name the 3 types of RNA and describe their function in protein synthesis. The 3 types of RNA are mRNA or messenger RNA, tRNA or transfer RNA, and then rRNA or ribosomal RNA. Here are the functions. mRNA is transcribed from the DNA. It's going to leave the nucleus, and this is the instructions for building proteins. It is going to work together with the tRNA, and the tRNA's function is going to be part of that translation process. It's going to pair up with the mRNA, and it's going to bring in amino acids to help build those proteins. So it binds with the mRNA at the ribosomes. Those codons are on, the anticodons are on the transfer RNA, while the codon for a specific amino acid are on the mRNA. rRNA, the main function is going to be part of the ribosomes. They are a temporary binding site for tRNA and mRNA. So let's look at this picture. Here is the process of translation. We have the mRNA here. That mRNA has a specific code on it. That code is going to work with the tRNA, who also has a specific code. This specific code codes for a specific amino acid. So UUU is going to code for the amino acid of lysine. So messenger RNA is going to bond with tRNA to bring in those amino acids at the ribosome. So this rRNA is here for that binding site. Use the text boxes to label the following diagram of the central dogma of biology, that central dogma or central principle of biology. Using those words, protein, protein, RNA, DNA, transcription, replication, translation. Here's how you should have labeled it. Replication happens up here in the nucleus of the cell. That's when DNA is making an exact copy of itself. DNA polymerase is that enzyme that is responsible for bringing in those those nitrogen bases. This is the DNA strand, so you should have labeled this as the DNA strand. Transcription is happening with that RNA polymerase. That is going to bring in the those nitrogen bases that are going to pair up with the DNA to create our RNA strand. And then translation is the process where the ribosomes are going to create that protein using that RNA strand as a template. Complete the following table describing the differences between RNA and DNA. Well, DNA is that information on how to create and maintain an organism. This is in the nucleus. And then RNA is an exact copy of the DNA, and it contains instructions for building proteins. The main difference between the two is DNA provides those instructions for the organism, but RNA has those instructions for building proteins. DNA is a double strand, RNA is a single strand. The bases used, DNA is going to be A, T, C, and G, adenine, thymine, cytosine, and guanine. And then RNA is going to be switched out for that T is going to be that uracil. Have those base pairing rules. Type of sugar used, DNA is going to be that deoxyribose, and RNA is going to be that ribose. What is the overall function of DNA and RNA? Multiple functions. They provide information of how an organism is put together and maintained. DNA is going to be the blueprint for the cell. It's going to provide those instructions, those main instructions for making proteins. The DNA is also a universal code based off of 4 nucleotides, which means that all codons code for the same amino acid. And RNA is going to determine which those specific proteins are based on that universal code. And then the relationship between DNA, amino acids, and proteins is that DNA calls for specific amino acids which are assembled into proteins. Below is a diagram showing DNA replication. Describe what is happening in each step. So the step 1, our protein building begins with DNA, and DNA is going to unwind or unzip by using that DNA helicase. Step 2, DNA polymerase is now, that's an enzyme, is going to now bring in complementary base pairs to pair up with the original DNA strands using that base pairing rule of A and T and C and G together. And then one DNA molecule plus another DNA molecule makes a replicated copy. They're an exact copy because of that base pairing rule. What characteristic of DNA replication is the most important in how genetic code is preserved from generation to generation? That genetic code is preserved from generation to generation because DNA follows base pairing rules. It has a universal code based on those 4 nucleotides, which means that all codons, so those codons are in that RNA, are going to code for the same amino acid. Different DNA sequences are going to be equal to different species. So we have a specific DNA sequence while my dog has a different DNA sequence. So differences in those sequences means that they're different species. Use the DNA template strand to create another DNA molecule and an mRNA molecule. So if I have sequence number 1, if I was going to replicate this for DNA replication, I would have GGC TAA. Transcription, I would have GGC UAA. Here are the rest of the answers. If you need more time, pause this video. Okay, we're going to be using this chart. Using this chart, we are going to answer the following questions. So what you're going to do is you're going to make sure that you read the code correctly. Here's the first letter, here is the second letter, and here is the third letter. What are these coding for? So 3 letters put together is one code, and the next 3 letters is another code. So I'm going to take UUC, I will find it on my chart. figure out what amino acid this codes for, and then I will look at GGA and find out what those 3 letters combined code for. So let's look at UUC. I'm going to find U, and C are going to be PHE. And I'm going to do that same thing with all the rest of these. So here are the answers. I'm going to get PHE and GLY. GLY and LEU for the next ones, and then ARG and a stop for the last one. Question number 9, blank is the name of the process by which a polypeptide is created. In this process, blank leaves the nucleus, floats into the cytoplasm where a cell organelle constructed from rRNA and protein called a blank bind to it. This organelle helps to bind mRNA and tRNA together. Each new tRNA molecule arrives at the mRNA molecule to add a blank in the growing polypeptide chain. A sequence of 3 bases on the mRNA is called a blank, and it is matched with the blank on the tRNA, which determines which amino acids are added. After this process is completed, the ribosome detaches itself and polypeptide continues on its journey to become a mature protein. Here's how you would fill this in to make it make sense. Translation is the process where a polypeptide is created. mRNA is going to leave the nucleus and float into that cytoplasm where it's going to combine with that organelle that's constructed from rRNA and protein called the ribosome. Each new tRNA molecule arrives at the mRNA molecule to add an amino acid to that growing polypeptide chain, and then the sequence of 3 bases on the mRNA is called a codon, and it's matched with the anticodon from the tRNA. If you have any questions about this review sheet and about this test, please contact me by email. I would be happy to help you out. Remember, our goal for this test is to be able to get a 70% or above. And to do that, you must study this information plus other information. If you are struggling with the information given here, make sure you research other information as well using the content information. Again, if you have any questions, feel free to contact me.